Yes. If you take away anything from the talk today about medical imaging and machine learning, is that a large sample size and reproducible methods do not mean that the methods generalize in practice. So, in fact, I think in our field we have lots of over-optimistic results. Okay, so what kind of things are we talking about? This doesn't work. We're looking at different types of images that may be produced in hospitals for various treatments or for prospective studies. So different modalities and dimensionalities of images. And there are many different tasks due to the volume of images produced, where we might want to use algorithms. For example, enhancing the quality of the image, to maybe outlining some specific parts of it to measure them, for example, to measure tumors, their size, to classification or diagnosis. So we are essentially taking some scans of a patient and then categorizing them into disease or healthy. So, for example, predicting cancer or dementia. And the steps for this, and I'm going to be talking primarily about this diagnosis part because this is the most overhyped part of the field. How this approximately works is that we first need to have a data set. And if we want the algorithm to generalize to some patients in the future, we need to have a large data set that sort of covers different types of patient and not only the specific ones we have in our study. And historically, these data sets for diagnosis were private data sets. So usually you would be in a collaboration with some experts who would have internal data from the hospital, maybe in the order of hundreds of patients. But more recently, I would say the last 10 years or so, public data sets are gaining popularity where people get together and release anonymized images, but usually you would have a set of images with the corresponding diagnosis. And then if you're lucky, maybe you have some metadata about the patients, like their age. That often not the case. And so with these kind of data sets, we now have some larger image data sets with maybe tens or hundreds of thousands of images, compared to applications like in other computer vision. There, the sample size is still much larger. And maybe to mention that I think the rise in the image dataset size comes a bit together with more complex methods which need more data. But there's nobody actually going through these hundreds of thousands of images. These datasets are kind of scraped from databases where you associate, for example, an X-ray that a person had taken with some automatic labels extracted from the radiology report. So just to exemplify here some typical datasets people use for benchmarking. This table is called large open source datasets. And we can see that 298 is already considered large by some people, and the largest are going up to 65,000 or so. So these are the number of patients that are in the data. Once we've chosen a data set, we can do our favorite machine learning model. So there's many different types of these, but essentially what all of them are doing are trying to find a mapping between the inputs and the outputs that you provided. So for example, images with a healthy or disease diagnosis. Once you've trained it to do this mapping, you can of course take a different image from a hypothetical future patient and predict a similar type of output for that image. But for training such an algorithm, we want to also know how well we are doing. So first thing we need is a metric. For example, sensitivity or specificity. We want to see how many of the cancer patients did we actually correctly detect cancer, and how many were false positives, for example. So of course, we need to evaluate this on some data that our algorithm hasn't seen, ideally. But often in practice, this is not data from the future. This is part of your existing data. You just split it up into parts, and then you do the training on one part, and evaluating on another part, and then you shuffle them a little bit and you do it again, so you have multiple results. So this is what is called cross-validation. I'll mention that again later. This is approximately how it looks like. So you train and then you test, you train, you test. And ideally, you would have some kind of external test afterwards, but this is often overlooked for reasons of publication. But if you've done this, you have some metric, you have some variability for that metric, and then you can compare it to, let's say, experts who you've also maybe shown images and had them guess the diagnosis, or some other algorithms, which is a common thing to do. And then you produce a table like this. So in this case, we're diagnosing skin lesions, and we see here 3 different data sets. And then there's a bunch of methods and 4 different metrics where we see some variability expressed. And then usually the method at the bottom is the one that's ours. You highlight it in bold because it has the highest performance. And then depending on the kind of lab you're from or tech company, you might claim that we should stop training radiologists. This was from 2017. All the radiologists I know are still pretty busy people. If you're not from a famous place, maybe you will write that you're humbled on LinkedIn that you have a new publication. But all in all, there's kind of there's a limited translation of all of these methods into practice, among others due to lack of external validation. So I want to highlight 2 examples why this happens, and maybe I'll focus mostly on the first one because it's the one that's maybe the most surprising. You can of course see how in this process we can do all sorts of methodological things that make our results better than they actually are. But maybe the main point is that large data sets do not solve everything, and it's indeed possible, even with a very large data set, to have a correctly done method which has high performance, but the algorithm is still doing something not what you expected. So this is an example with skin lesion classification, where I think these algorithms were reported to be better than dermatologists at detecting melanoma. But what the researchers in this follow-up paper showed is that the algorithm has actually memorized a spurious correlation between what's in the image and the diagnosis, and the correlation was with these pen marks, which dermatologists sometimes place around the skin lesion. So how we showed this was that this is actually a healthy skin lesion. The network is predicting it correctly by itself with high probability and confidence that it's healthy. But if the pen marks are added and you have the same picture, then the decision completely flips. So just because there's a correlation in the large data set between pen marks and melanoma, that's kind of what the network has learned to look at. And there's lots of other examples that me and Amelia have been collecting. So another interesting one is the chest drains, which are inserted into the lungs for people suffering from a collapsed lung. So if you see in a chest X-ray that there is a chest drain inserted, you don't need to diagnose the person with a collapsed lung. It means it's happened already. They had the drain inserted. So then if you take away actually those examples from the data set, then the performance where higher is better and one is perfect, the performance drops actually quite a lot. We can figure out that you have severe COVID if you're not able to stand up, which we can see from your shoulders and sort of. And it gets worse because these are kind of things we can see when we inspect the images, and these confounders are something we can relate, like the pen marks or the chest raise, facial position. But the networks appear to be memorizing more things which we cannot really figure out what's happening. So in this study, rather than predicting the diagnosis from chest X-rays, they tried to see, can we predict the ethnicity of the patient? And apparently we can do this quite well, even though radiologists say that they cannot do it if BMI has been accounted for. So we can predict this with, from the original images, we can predict this almost perfectly. But what is even more surprising, if we degrade the images by filtering them, so this corresponds to the performance over here. There's almost no drop in the original performance. So there's some correlation that we learned that's still present in images like this, and the diagnosis, and the race of the patient. So it's likely correlations between like where different demographic groups go to the hospital, and if one demographic group is more often in the hospital with a Siemens scanner, and the other demographic group is more often in the hospital with a Philips scanner, that's the kind of patterns we could be memorizing here. And a similar result in dermatology again. In blue, we're predicting melanoma again, so 100 is the best. You can do it, I can say, with 90% correctness. If we start blocking out the actual skin lesion, okay, the performance drops a little bit, but it's still much higher than random. And again, here it could be things like the skin color, or whether certain tools were used in the diagnosis. So for example, here you see these corners that indicates dermoscope. And we don't actually know exactly why, what is being memorized. So performance can be high even if the network is not looking at useful information. How am I for time?
We still have 7 minutes left.
Okay, and things appear, this is often not addressed, and it seems that kind of things are going well because people share their code and people use public datasets. This is an example from a recent conference, and you can even read all the conference reviews. So there's a kind of illusion created of reproducible results. In some cases, they are, in fact, actually reproducible, not very often, because, for example, some files are missing from the repo to be able to rerun the code. But Attila has taken it upon himself to actually document for a conference all of these things. And some repositories we can, in fact, reproduce. But it's not really clear, even if the same numbers come out, whether these results are useful. In part, as we've seen, because the data set might be problematic. In part because it's not clear if the reported result is actually better than other results we've had. So there's all sorts of problems with if people test statistical significance of the difference, which doesn't happen very often. Usually it's done in a way that is wrong and inflates, and you can arbitrarily inflate the significance. Even if this is done correctly, then it's still not clear what is a kind of practically useful difference between algorithms, because two results can be very close, can be statistically significantly different, but very close together. And for this, we looked at a platform called Kaggle, which is where people compete basically for the best results on different data sets. So usually somebody organizes a competition, they release some data, and they offer some prize money. So this was in 2017, solving lung cancer, and if you are the best, you will get $1 million. And almost 2,000 teams signed up to do this and submit their algorithms on this data set. And then how it works is that there are two sets of data, the public data and the private data. You use the public data to optimize your method. Then you know how well you're doing compared to other competitors. At some point, the competition closes, and they evaluate it on the private set. And the ranking also changes. So in this case, you can see that the team who was the first winner on the private set, GERT 1, 2, 3, was actually in place 136 on the public set, but suddenly became the winner. And I won't go into all the details of this, but basically here in blue, we show how much the ranking of an algorithm can change if we switch these data sets. And in brown, we show how different are the best algorithm and the one that's 10% down the leaderboard. So in that case, number 1 and number 200. And basically, we see that in many places, that improvement is actually much smaller than the expected variability you'd expect if you had taken a different set of data. And something that was really cool here is that at submission of the paper, we only had 4 of these medical data sets on Kaggle. And the reviewers had asked why, because there are much more, but that was already a year after. And we reran the analysis on the 4 that were released after we submitted, and it was even worse. So, yeah, of course, I haven't talked yet about that detecting something in retrospective patients doesn't mean that the more patients will be treated better. Tiny self-promotion, if you find this interesting, we have a webinar series. I will share the slides so you can get the links, but we talk about more surprising things we find in datasets. And that brings me to my summary. There are some promises, but the algorithms that are being produced are not very robust and generalizable. And I think we should do more critical papers about datasets, reviews, and evaluation. Yesterday, I got the idea of maybe having a course on spotting errors and better incentives to do all of this. Thank you very much.
Thank you.